Hi, everyone. We are so happy to have you, whether you are joining us here today or watching later on our Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already, and include any questions you might have in the chat box below. We will have time to answer questions at the end of the presentation. My name is Monica Joanna Elnacave, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants. By partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trials. Today, we welcome Dr. Zachary Grinspan, who recently joined us at our Cure GABA-A Scientific Roundtable. Dr. Grinspan is also listed under the Cure GABA-A website as one of our recommended doctors. Dr. Grinspan is the Chief of Child Neurology and the Director of the Pediatric Epilepsy Program at Cornell Medicine and the NewYork-Presbyterian Komansky Children's Hospital. His expertise expands to translational research and clinical informatics. Join us today in exploring Dr. Grinspan's groundbreaking collaborations, which include over 100 published research articles spanning various disciplines. He is at the forefront of national pediatric epilepsy research, leading multi-centered projects. In this session, Dr. Grinspan will explain the details of the Ravicti Clinical Trial, focusing on the selected GABA-A variant mutations. This choice is informed by a study conducted by Dr. Katty Kang from the Kang Lab at Vanderbilt University, which affirmed the positive impact of Ravicti on individual cell models with alpha-1 and GABA-2. I'm handing it off to you, Dr. Grinspan. Please share with us your presentation of the what, why, how of clinical trial on Ravicti with our Cure GABA-A community.
Thank you so much, Monica, and it's a pleasure to speak with you all today. This was sort of a whirlwind. Monica, I met you in Orlando, this amazing work that Katty Kang has done. And so it's a pleasure to talk with you all today, to talk about our work so far and why we're very excited to extend this trial to the GABA-A community. So what I want to talk to everyone about today is about this clinical trial that we did using a medicine called phenylbutyrate for two rare disorders similar to GABA-A mutation disorders, one called STXBP1 and the other SLC6A1. And the motivation for my being here is Dr. Kang's recently published work where she found that this molecule 4-phenylbutyrate helped improve several outcomes in a preclinical model of a GABA-A mutation. And this particular work is sort of the tip of the iceberg of what she's been working on and discovering. But it's very exciting, and we're motivated to act on it. We currently have a clinical trial using this molecule for these two disorders. This is just showing you the clinical trials website. And the funding for this study that we've done so far has been really amazing. We got launched with generous funding from SLC6A1 Connect, the Orphan Disease Center, from the STXBP1 Disorders Foundation. There was a Lulu's crew, Team STXBP1 did a bike ride to raise money for this. Clara Inspired was a private foundation that helped fund us. And then the medicine, phenylbutyrate itself, Horizon Therapeutics provided that to us for the first 20 patients at no cost. They have since upped the ante. They've provided us medicine for another 30 patients and have also provided some research funding. Horizon is now Amgen, so I probably should put their logo here. And then the work that I share with you today and the ongoing study is a collaboration with Children's Hospital of Colorado. My partner there is Scott Demarest. Speaking of which, this is our team here at Cornell and at Colorado. The thing I would really highlight is that it's a multidisciplinary team. Jackie Buret is a basic scientist, and then myself on the clinical side. We have experts in developmental medicine, in neurogenetics. We have folks helping us out in the lab. And then at Children's Hospital of Colorado, he has his amazing team as well. So let me talk briefly about these two other disorders. On this call, I think they're valuable to give some summary because there's similarities to some of the children who have GABA-A receptor mutations. STXBP1 encephalopathy is rare. It's about 1 in 30,000 live births. Most of those children, but not all, have epilepsy. Intellectual disability is largely there, 88%. It's either severe or profound. Some have autistic features, some have hypotonia or movement disorders. The STXBP1 protein is this little peanut here that is involved in synaptic vesicle release. So this is the synapse, and this is one of the vesicles that would have some neurotransmitter in there. And then this protein, STXBP1, the blue, helps make that synapse, that synaptic vesicle rather, open up and release its contents. The other disorder, SLC6A1, is similarly rare, 1 in 35,000 live births. Most of those children also have developmental delay, low tone, intellectual disability. Many, but not all, have seizures. Movement disorders are common, as is autism, and attention deficit. This protein is also involved at the synapse. It's GAT1. It's a GABA transporter, and it's this coffee bean here that's at the nerve terminal. It's there at the end of the neuron, and it's also there in the glial cells. Two labs, my colleague Jackie Buret's lab here at Cornell and Dr. Kang's lab in Nashville at Vanderbilt, both had this idea to use a group of molecules called chemical chaperones for these two disorders. MUNC18 here is the same as STXBP1. In Dr. Buret's case, the story is that her husband is a cystic fibrosis researcher, and that community had been very interested in trying to rescue some of the misfolded proteins in cystic fibrosis. Her idea was, well, maybe this will work for neurologic disorders. Both of these papers showed effect of this molecule, phenylbutyrate, for the respective disorders. And then most recently, just in the fall of 2023, Dr. Kang showed similarly in one GABA-A mutation in a mouse that this molecule also helped in GABA-A mutations. So what about the molecule itself for phenylbutyrate? This is a repurposed drug. The FDA has already signed off that this drug for phenylbutyrate is effective for a group of disorders called urea cycle disorders. To make a long story short, in the periphery, in the body, this molecule helps gather extra ammonia and helps the body to remove it through the urine. And so the molecule, I think, was approved in maybe the 90s or the 2000s, has been part of standard therapy for this rare group of disorders. What's nice for us in the neurology community is that the molecule crosses the blood-brain barrier. The original molecule that was approved is called sodium phenylbutyrate. Here it is, and it's super disgusting. It's bitter, it's salty, and when I've talked to the metabolic doctors that use it, what they say is that people kind of hold their nose and they stomach it, and they're able to take it largely because these are sort of neurotypical children and adults, and they know that if they tolerate the nasty tasting medicine, that their other symptoms will improve. Because of the foul taste, the pharmaceutical industry sought to come up with a better version of it that would be more tolerable, and they succeeded. So Ravicti, the drug we're talking about, is glycerol phenylbutyrate. What it is this is a glycerol backbone, and you've got 3 of these phenylbutyrates kind of glommed on there. So what happens is you swallow the liquid, and then in the stomach, the phenylbutyrates pop off and are digested, and then you have them available in the blood, and for us in neurology, in the brain. We don't know why this molecule works. There's several different theories, and if you look through the papers, each paper is sort of wed to a different idea of how this molecule is working. I'll go through some of the concepts, although I don't know which one of these is correct. One idea is improved trafficking, i.e., that either it helps the misfolded proteins get to where they're supposed to go, or it helps the normal proteins get to where they're supposed to go. The end result being that you have more functioning protein at the cell membrane. There's another idea that it reduces aggregates. Sometimes if you have misfolded proteins, they glom together and they make these aggregates of proteins, and those aggregates may have other toxic effects. And so if you can clear out those aggregates, then maybe the cells work better. A third idea is that maybe it makes the proteins work better. Either the mutants or the wild type, that somehow the molecule supports the function of the protein, and that it improves their efficacy, improves the molecules working the way they're supposed to. In the SLC6A1 community, there's some evidence that phenylbutyrate directly improves the function of that mutated protein, of the wild type version of the mutated protein. There's this other idea that it reduces endoplasmic reticulum stress. The cells have this natural alert that gets fired if there's a lot of unfolded proteins, or if the endoplasmic reticulum, which is responsible for sort of taking long strips of amino acids and folding them into proteins. If that system gets stressed out, there's a stress response that can be toxic to cells, and sometimes cell death can result. And so chemical chaperones as a class may lower this stress, and that may also have therapeutic effects. And finally, there's this idea of histone deacetylase inhibition. The concept is that you have these at the chromosomal level, the DNA is all wound up to store it, and it will unwind from time to time to let different proteins get transcribed and expressed. The histone deacetylase inhibitors help open up sections of DNA to allow those proteins to get transcribed. And so that's one action that people look at with phenylbutyrate to think that maybe that could be why it's helping. Regardless, we were very moved by the clinical effect, and so we launched a trial. The initial trial, these are the inclusion and exclusion criteria for the initial trial. For STXBP1, we thought, listen, almost all of these children have seizures, so we want to focus on that. For SLC6A1, some children have seizures, some don't. And so at that point, we were broader with our inclusion criteria for SLC6A1. And we've always preferred younger children. I should note that these inclusion criteria have changed for the trial going forward. It turns out that Ravicti is good down to the day of birth. And so going forward, we can enroll newborns. And then second, the effect that we saw on seizures for both of these populations was very good for trial design. We know that most of these communities where there's a neurologic mutation have a broad range of symptoms. Some children have seizures, some don't. Some have developmental delay at different degrees. And when we talk to families, it's clear that things like development, communication, relatedness, and things like that, behaviors, are so important to families. From a trialist's standpoint, those things are hard to measure and hard to count. So we focused on seizures in the trial because it helps us show an effect. There are a handful of exclusion criteria that are listed here. We have not needed there's been no child that we've thought about enrolling that has had a problem with one of these. The drug itself, we've been very pleased with its safety and tolerability profile. Here on the screen is listed the known side effects and drug interactions that are already published, and the FDA has already signed off on that these are like reasonable side effects for an FDA-approved medicine. The 4 that are highlighted in red are the ones that we've seen in the trial. Decreased appetite, this happens not uncommonly. There's a sweet body odor. Some families have said that it's sort of almost a curiosity. It doesn't seem to be toxic or negative. It's been described as honey-like. It's been described as wildflowers in a hot car. A few families have said that it's a little bit gross, but just a little bit. I don't want to go, this has not been dose limiting. Maybe I should put it that way. The third side effect, metabolic acidosis, is probably the most serious side effect that we've seen. What can happen is that phenylbutyrate turns to phenyl acetate. That's an acid, and so it can lower the pH of the blood, and then the body has to account for that. The kidneys are very good at that, but in sick children or children who are medically tenuous, that acidosis can be too much to manage. We had one child for whom metabolic acidosis became a problem. The child became quite nauseous, couldn't eat anything, ended up going to the hospital. We then disenrolled that child, so we took her out of the trial. What's interesting is that that child has since re-enrolled because Ravicti is really sort of the only thing that worked for her. Metabolic acidosis is something that in a neurology clinic we manage regularly. If anyone's had a child on the ketogenic diet or knows anyone in the community with a child on the ketogenic diet, acidosis is common for that. And we have a variety of medicines to help us monitor it and control it. Anything from like baking soda to a medicine called Bicitra. Somnolence was a problem early on in the trial. We were very aggressive for some of the early kids and titrating up to the maximum dose very quickly, and many children got super sleepy. We have since learned that if you slow it down and titrate up to the maximum dose over a few weeks instead of a few days, that the sleepiness doesn't seem to be as much of a problem. Outside of the trial, we have one young woman who has had some menstrual dysfunction. We haven't seen the rest of these. As far as drug interactions, phenylbutyrate is listed to potentially reduce the effectiveness of midazolam. We worried about that at a high level because if someone is seizing and someone uses midazolam, that maybe won't work as well. This hasn't been a problem thus far in practice. Here's some of the things that we are monitoring. Seizure burden has been the outcome where we have seen the clearest effect, and so that's really where we've focused. We are also measuring quality of life, communication, sleep. We're doing some development work with both Vineland, and some children get the Bailey as well. And we've been very interested in EEG abnormalities. Behavior is clearly important for many communities. The current trial that is essentially completed, we sort of started thinking about this measure called the ABC. It's the Aberrant Behavior Checklist. We've since incorporated that into things going forward. And then many children have abnormal movements. We have thought about how to measure that, but have found it in practice difficult with our current trial design. So here's how it works. There's an initial screening call. And actually for many families, sometimes there's even a call before that where people are just looking for more information about the study, which we're also happy to do. But officially, like once you're in, we do a screening call to find out whether you're a good candidate for the trial. We run through those inclusion-exclusion criteria. Then you're enrolled, and we do 4 to 5 weeks of weekly telehealth visits. The purpose of those visits is to get to know you and your child. We find out about how often you're having seizures, what medicines have worked, what medicines haven't worked. We gather MRI data, EEG data, and just take a thorough history. As a medical student, and as a resident, I was always under the illusion that you could just get to know somebody at a single visit, and maybe in an hour or 90 minutes, you would kind of know everyone's story. But in practice, that's rarely true. So this has been nice because it's allowed us to review certain pieces of data. If there's an MRI or an EEG that the family hasn't been able to get, we can sort of apply a gentle reminder like, okay, we just need this one piece of data beforehand. So this has worked out really, really well. The other reason that we do so many visits is that in a pilot study like ours, we have this objective stuff like how many seizures did you have, but we're very interested in the qualitative. We're very interested in what is your experience, what is it like, what are the things that make your life hard? We want to know about that because as we get into, once we apply the drug, we want to know if it's had any effect. Some of the measurements that we apply are good for the things that the drug does, but not always. And so the qualitative stuff helps us a lot. The first, after those telehealth visits, you and your child would then come to either New York or to Children's Hospital Colorado outside of Denver. They are funded, but we're still getting them up and running for this portion. So currently, we're just enrolling in New York. This is a 3-day, 2-night evaluation. What happens is often we do like the developmental visit that first day, and then the child is admitted to our epilepsy monitoring unit for overnight EEG. On day 2 of that visit, we get some labs. I think we do an EKG. There's a skin biopsy that is part of the protocol. Rather, let me reframe that. There's a skin biopsy that's part of a parallel protocol that's not required. We have a neurogenetics program here as well, and they have an open protocol that any genetic epilepsy, we're able to get a skin biopsy so that the basic scientists in that group have populations to work on. And so as part of the visit, we do a skin biopsy to get them the sample that they need. And then the drug, the first dose is given sort of late morning of that second day. We start with a very small dose. It's 1/9 of the goal dose. And as a quick anecdote, the reason we did that was actually because of a parent. In the STXBP1 community, John Oldenhoff is a parent of a child with STXBP1. He's on their board. He also professionally designs phase 1 studies for industry. And so because STXBP1 was the first genetic community that we did this trial with, he was very involved in the study design and made all of these suggestions based on his expertise. And so largely based on his advice, we decided to use a small dose in a hospital setting to make sure that there weren't any emergent or urgent adverse effects that would land someone in an ICU that would cause status epilepticus or something like that. And so that's really been the justification for doing this initial exposure on the inpatient side. So the first 3 doses are given inpatient. You get 2 on day 2 and then 1 in the morning of day 3. And then we come up with often like a paper bag with, full of bottles of Ravicti for you to take home. You're then home for 12 weeks. We ramp up to the goal dose, which takes 2 to 3 weeks, depending on how well your child tolerates it. And we're in touch every other week, again, by telehealth. We have specific things that we're measuring. Millie Stone, who's our research coordinator, sends surveys and things to fill out. So we're collecting all this data. And then the conversations with me are understanding the experience. We've heard, for example, there are some families where they say things like, my child is more engaged. And I don't think any of our specific measurement tools are going to pick that up, but having that qualitative response is really helpful because it helps us design new measures. It helps us find different measures for more definitive trials. And again, it helps me to get to know all of the details of how your child is tolerating the medicine. After 12 weeks, you come back either to New York or to Denver. And then we have 2 weeks of follow-up, and then there's sort of two paths. If you are done, thank you so much, this didn't work. I'm glad that I could participate. That's okay. We then wean off the medicine slowly and stop. We do have this extended use option, meaning that if you want to stay on the drug, our contract says we can continue to give it in this extended use capacity through December of 2025. During that period, we would do quarterly visits, 3 of those by telehealth, and then once a year, we do want people to come back so we can do a physical exam and then get a routine EEG here, and then do a developmental assessment. December 2025 is very specific. The reason that we chose that is that the exclusivity that Horizon, now Amgen, has runs out in the second half of 2025. So there's this anticipation of generics coming onto the market. Horizon's attitude seemed to be that they would sort of be more than happy to pass this off to generics. We're still learning about what Amgen's plans are for the drug. Hopefully I'll learn about that over the next several weeks. But this is the promise that through 2025, December, we can continue to give it. Our experience with insurance is that for many, but not all children, we have been able to get this approved. There's one child who just by virtue of a letter from me, I essentially said, this child had a lot of seizures. We started this drug, the seizures went away. That was enough to get insurance to cover it. We found that many commercial payers are willing to listen to us for children not in the study. There's another formulation of phenylbutyrate called Feburane. It's a taste-masked sodium phenylbutyrate. We've been able to write that for some kids outside of the trial as well. For the managed Medicaid, for the publicly insured, we have not had as much luck. What we believe is that as we publish our findings and as we get this listed in compendia, in these online descriptions of on and off label uses of different medications, that getting insurance to approve this after 2025 will be easier. Let me show you some preliminary results from our study so far. My point in showing this to you is really to share with you the excitement that we have. I also should be cautious in that this excitement, in the epilepsy community, we've been here before. There have been several examples where early results have been very, very promising and they haven't panned out. So I'll share with you these promising results to give you a sense of where our enthusiasm is coming from. But I want to temper that with the recognition that this is early, that these are a handful of cases, and that we're still learning about the value of this drug going forward. So safety and tolerability. As I said before, we've been very, very pleased that we have not seen new side effects. The FDA has a bunch of side effects in their listing, and everything we've seen has been in line with that. And then the drug is pretty well tolerated. We really aren't seeing a lot of missed doses. The kids just kind of slurp it down. It's a liquid. It doesn't taste bad. Here's an example of a child that did very well. This is an 11-year-old boy with an STXBP1 truncation mutation. At baseline, he had a handful of seizures a week, often in clusters, but not always. He had a cluster of seizures on his trip up to New York, and then was essentially seizure-free for several weeks. He had one breakthrough here during our study period. So this family was very happy. And then this is from SLC 6A1. I affectionately call this child our respondingest responder. So this is sort of the best that we've seen. This is a child who was having 6 or more absent seizures a day. And when I say 6 or more, it was probably hundreds. He came up, we titrated the drug up slowly. He had a handful, and then has essentially been seizure-free. I just did my 2-year follow-up with this child. He is still seizure-free. This is a summary of the 20 kids in the first group with their 10-week outcomes. Green are responders, yellow are indeterminate, where they're better, but it's not clear if it was from us or from something else, or they're not quite better enough. There were 2 definite non-responders. And then in the SLC 6A1 group, there were 4 children who really did not have seizures. So 11 out of these 20, 50% or more seizure reduction, where as a clinician, I felt good saying, yeah, I think it's from the drug. These 3 indeterminate, there was one that sort of had a reduction, actually 2 had fewer seizures, but it wasn't quite enough. And then there was another kid who is having lots of epileptic spasms. The mom is convinced that the number of seizure clusters are less, but it's been really hard to measure that. These 2 non-responders both had epileptic spasms, infantile spasms, or infantile epileptic spasm syndrome. And it just didn't do anything. There was one child where this child is still having spasms. And then another child, we enrolled the child when she was still having spasms. The spasms went away in our observation period and essentially never came back. That child has really not had seizures come back. So we're kind of on the fence of whether it's done good, but we can't say that it's been a response. I want to share with you another exciting thing about the study, specifically for SLC 6A1, which is that we have a biomarker for that disorder. A biomarker is some disease-related measurement that helps you track whether a drug is working or not and tracks with clinical response. I don't know how many EEGers are on the call, but as a brief primer, this area on the screen with all of this high amplitude stuff and these little embedded spikes is an absent seizure. This figure on the bottom is called a compressed spectral array. It essentially compresses this EEG information. Each one of these little yellow spikes is one of these seizures, and this is 1 hour's worth of data. So this child was having dozens of seizures an hour. When he came back 6 weeks later on the drug, his EEG had cleaned up. This is a sample EEG with none of those spike waves, and here's that compressed spectral array with none of those spikes. Take everything I say with a grain of salt. This is an open study. I said this in Orlando, and I'll say this to those of listening. I know that I'm biased. I'm a physician and a clinician at heart, and all I want is for my patients to do well. And that biases me in an open label study because that I want them to do well. So that's a limitation, and I can't make that go away. In the SLC 6A1 group, there are some kids who have these staring spells and their eyes flutter. And my colleague Scott Demarest and I go back and forth with some of them. We're like, maybe they're seizures, maybe they're not. We're not sure. Regardless, absence seizures are not countable. It's nice to have that EEG because that kind of makes it clear that there's been an improvement. But absence and staring spells can be challenging to measure. And in the STXBP1 group, many of them have seizures at night. They have tonic seizures at night. Many children with like a Lennox-Gastaut syndrome clinical picture have this as a common seizure manifestation. And you always wonder, are we actually counting them all? We rely on parent reports. We rely on parents to be accurate and do the best they can, but we know that there's always wishful thinking, and that may affect our counts. So what are we doing? We still have to get our first paper out. That's really on me. Horizon, now Amgen, has given us this medicine for 30 new patients. That's why I'm talking to y'all today. We are enthusiastic about what other disease entities this might work for. We've given this to 5 or 6 children with SYNGAP. We're excited to work with GABA-A receptors. And then there are a handful of other disorders where we've been in conversations to figure out whether it's worth a try. And then that EEG biomarker, we're still working to quantify. So that is the end of my prepared remarks. But I am really yours for questions. I do want to end with a thank you. This trial, this whole idea was really launched by parents and by advocates. I think Amber is on the phone, and she will certainly back me up. There is nothing quite like being on a research call where there's a parent advocate on that call. It lights a fire like nothing else. And so those fires really drove this, and the seed funding really drove this. And now, we're in a position where we've got more solid funding from pharma to keep it going. But we're enthusiastic, and I'm just very excited and glad to be speaking with y'all from GABA-A today. Thank you.
Thank you so much, Dr. Grinspan. And I also want to note that Amber Freed is on the board of Cure GABA-A. She is our Chief Operating Officer, and you're right, we are so lucky to have her. I have some questions that came in, and as more come in, I'll read them off.
Great.
But we have one from a previous discussion that I had with a mom about the drug, and it's something that you touched on actually today. But if you could expand more about whether the sodium, the glycerol, and I'm going to add the taste masked phenylbutyrates, if they all work the same.
Great question. So let's see. Yeah, so here are the 2 formulations on the screen. This is the sodium phenylbutyrate and the glycerol. One child who I prescribed the plain old sodium phenylbutyrate, it's a powder. That was a very disabled child with STXBP1 who had a G-tube. And so the taste was sort of irrelevant for that family. I want to be very careful with what I say because I don't want to give false hope, but that family is so happy. The child was having lots and lots of seizures, and those have really stopped. The mother thinks that the child is seeing better. And it's a good example of where like it stopped me in my tracks in clinic because it was such a dramatic claim. The clinician in me was jumping up and down, and the scientist in me was saying, you just can't believe that. You can't actually, like, it's too unbelievable. So I say that as a clinical anecdote and as the clinician, not as the scientist. But it does give me some confidence that, phenylbutyrate is the active molecule. It doesn't really matter how you get it in, whether it's sodium or glycerol. The taste masked product, the Feburane, they have not funded me. All the funding is from the Ravicti folks. We've had some luck getting that prescribed. It's clearly more tolerable than the normal sodium phenylbutyrate. And I don't yet have an anecdote of it working, but it's just because I haven't given it to enough kids when we're kind of still working on the insurance. The early reports from one family, we started to see some of the side effects. So we knew that it was getting in and she was tolerating it, but we're still waiting to see if it has benefit. So my larger approach is that in the study, I have Ravicti. I can carefully select children to be in the study, and we can do all of this formal measurement to help us with publications. Out of the study, the doctor and me, like, this is a nice idea. I can just write for it. It's not hard. It's just write a prescription and it's safe. The issue is always the cost. It seems like commercial insurers are more willing to kind of go on a limb and do what they can. The Medicaid is harder because they have more regulations and it's just stricter. The Feburane is priced competitively, and so that's often an easier pitch to these payers. I haven't said this yet, but Ravicti is $800,000 a year, and I saw one of the families shared with me, like the letter they got from the insurance company, and the conversation went like this. Your doctor has prescribed phenylbutyrate, Ravicti. This will cost $100,000 for 1 month's supply. And then the insurer goes back and says, okay, we're going to pay $48,000 for 1 month's supply. And then, that's agreed on. And the copay to this particular family was $75. So that was sort of an example of where, there's this exorbitant exchange of dollars that the family was protected from. We've had other families that have gotten bills, that are like $10,000, and they're like, we'll stay on the study supply. We don't want to go to insurance. So the cost has made the Ravicti difficult outside of the trial, but not impossible. That was a long-winded response. I hope
that's
that was
okay, because it's wonderful to hear everything you have to say. And you're right on that I also saw it's one of the top 10 most expensive drugs in the US.
Yeah.
Here's a follow-up question. Is it dose dependent, and how long does it live in the system?
Yeah, that's a great question. Let me answer the question clinically rather than as I'm not a pharmacologist, but here's what we've seen clinically. There are a couple of kids where when we get to the maximum dose, which is what the FDA has set as safe, we're not pushing past the FDA limits, we get seizure control. I have one kid in particular where he had side effects. I think he got sleepy or he stopped eating. So we backed off the dose to maybe 60 or 70% of the maximum. His appetite came back, but so did his seizures. And then we slowly titrated back up to the goal dose. The seizures went away and his appetite stayed. So we were able actually to get him back into control without incurring new side effects and actually managing the side effects. Which interesting, I think it's the same kid. He grew. And so mom called me and it's like, oh, the seizures are back. I'm like, well, how much does he weigh now? And he had grown a couple inches and he was a little heavier. So we dose adjusted. We got him to the new dose and we got control of the seizures again. So that's one example where my instinct is that you really need that maximum dose to get there in order to get enough of a concentration in the spinal fluid to have the effect. The other thing is that we've had a few families play around with twice-a-day dosing with mixed results. There was one family that went to twice a day and the seizures really came back. And so that family went back to 3 times a day dosing and the kid did okay. And then I have another family that successfully did go to twice a day dosing without sacrificing seizure control. So, I'd be nervous to go to once a day dosing because I don't want the seizures to come back, but twice a day dosing may work for some children.
I'm actually going to jump down to another question because it's a great segue. You mentioned, this is from Mia, sorry, Mia. You mentioned titrating up in our group. Will all patients be titrating at the same speed in the trial?
Yeah, good question. So we have a schedule. It's like 19 days, and that's sort of where we came up to. That is intended to be our first kind of like a straw man. It's like, let's try this and see how it goes. Families regularly contact me during that period and say, my child is too sleepy, my child stopped eating, and then we stop, we back off, and then the goal is to get to the idea is to get to the goal dose, ideally by like 4, 5, 6 weeks, so that we have a good period of time in which you're at the goal dose. But we are in pilot study land. This is not a regulatory study, and so we have flexibility to titrate up as per your child's needs.
Okay, great. And Augustina wants to know, in the event of an adverse effect, are the medical costs covered by the insurance or the study?
Insurance. That's important. So there are multiple layers to that question, which I may need to address with that particular family.
Okay.
But in general, what we say is that insurance, it's an FDA-approved drug, so insurance covers all the admissions. And we did have one child that did have an adverse effect requiring hospitalization, and all of that was covered by insurance. I am available. In that child's case, I was regularly in contact with the local physicians, and I've had several other often close friends who have taken care of some of the kids in the trial who have reached out to me and called me and kind of talked through things. But to that specific question, insurance covers. Stepping back to sort of what gets paid for and what isn't. All of the telehealth visits with me go on the study. The drug goes on the study. We do an evaluation, a developmental evaluation that goes in the study. Actually, we have a physiatrist now who does an evaluation that goes on the study. The admission to the hospital for EEG goes to insurance. And the lab work often ends up going to the insurance. On that follow-up visit, the EEG we've ended up charging to insurance as well. So we've sort of had a mixed model of what gets paid for. Part of this was that we wanted to be good stewards of dollars raised by foundations, and then we kind of got a model. And now that we have pharma funding, we just sort of have stayed with the same model.
Okay, Ali wants to know, do children have to have seizures to enroll? Ali's not on the call right now, but her follow-up question would be, do movement disorders also make children eligible?
We are only enrolling children with seizures, and we are looking for children who have a lot of seizures. The reason is that it's been such a layup with the first 2 groups that we've seen these clear effects on seizures. It's very compelling to when I give a talk, when I talk to families. So seizures are what we want to measure in the context of the trial. We know that seizures are the tip of the iceberg for these disorders, and we have not forgotten about the importance of communication, development, behaviors, sleep. We measure all that because we know quality of life, because if we see that effect, we don't want to lose it. But in terms of kind of like selling this to the world, if we see an effect on seizures, it's kind of easier to make the pitch.
And I just wanted to mention that we are trying to onboard all of our Cure GABA-A community into Matrix and Citizen, but mostly Matrix, so that we can keep track of all of these things that you mentioned earlier that maybe, you can't keep track of during the study, like a child being more connected, eye contact, and so on. So I'll put the link in the description box below. And we are also trying to compile all of our community's EEGs and their tracings to determine a biomarker amongst our gene community.
That's outstanding. So for SLC6A1, that's been a huge win that I did not expect, and it's going to make this very much easier to pitch and to sell. We haven't figured out how to quantify the EEG, but to give you an example, for the SLC6A1 community, there's a natural history study that was conducted at UT Southwestern, and the physician reading the EEGs saw one of the patients who was in the trial and called the primary investigator and said, something changed. What did you guys do to this child? And it turns out they were in the study and they had gotten the drug, and it had very much improved the EEG, again, in that disorder. So this is sort of an example of a blinded visually apparent change. So we'll be interested if, for example, we see EEG changes in GABA-A and SYNGAP or other diseases. That becomes a very compelling measurement to try to pitch this for broader access. And just to underscore that, this is not regulatory, right? We've got an FDA-approved drug, so our audience is not the FDA. We have two audiences. It's the payers, because sometimes you send them a manuscript and it's enough. But there's also these 4 compendia that list all the drugs, that have this rigorous process where they evaluate what are reasonable on and off-label uses of these different medicines. Many payers use those as their trusted sources. And so if we were able to create sufficient evidence to get it listed in compendia, it then makes it easier to access for the community. And so more neurologists and geneticists could then prescribe it.
That's wonderful. And there's one last question, I'm sorry, and then we will wrap up. I know we're running out of time, but Michelle wants to know, do you need a prior EEG or MRI before being enrolled in the trial?
I've seen this happen where in order to get into a trial, you need an EEG. So it's like, all right, well, come visit me clinically and we'll get an EEG, and then we'll kind of know what's going on and have a better sense of whether we should enroll you or not. So like the absence of those things doesn't rule out enrollment. But I think it would be nice to have a sense of what the EEG looks like before formally going into the study.
Okay, great. Well, thank you for sharing all the information, Dr. Grinspan. It was so informative, and you are, as always, so well spoken. It is so easy to follow and understand what you're saying. And the enthusiasm behind the drug, I mean, I'm enthusiastic as well. So thank you so much for joining us here today. And if anyone has any follow-up questions, they can reach out to us at Cure GABA-A, and then we can forward them off to Dr. Greenspan.
Excuse me. Oh, sorry. We had a question. So
I'm so sorry. I didn't
go
see
ahead.
you.
Go ahead.
Yes, okay. So we are Crystal and Nicolas. We are from France, and we are the parents of Colin. We have a question. Our daughter, she has a lot of seizures, like every 5 to 10 minutes. She used to have that, and she had a lot of medication. Now she seems better, but the neuropediatrician told us we don't know how long it will last. So our first question will be, when you're going into the clinical trial, do you remove everything, every medication before going? Like, how do you deal with that?
No, so we're looking for children who are on medicine and still having seizures.
Okay.
But I'm glad you asked your question because there's two things I didn't say. One is we're really restricting to children in the United States. The reason is that in order to give me the drug, I had to promise up and down to the pharma company that I would only enroll children in the US because I'm not allowed to ship the drug out of the country. In the extended use period, the drug comes to Cornell, and then our like amazing research coordinator gets it into a box and ships it to you. But we would get in a lot of trouble if we shipped out of the country. This is not to say you don't have options. Depending on who your neurologist is, there's an international group of folks who have been paying attention to this.
Well, he's in link with Dr. Rinke. Yeah, he's in link with Dr. Rinke
amazing.
Yeah, and so he was in link with them asking Dr. Ortiz for the last drug we were supposed to add. At the end, they added CBD as a drug to treat, and that helped a lot for us, but we don't know how long that will last, like I mentioned. So he's already into sharing information, etc. So of course, like if we need to come for a few days, we will come to US, no problem. But as it's a long term, we understand. I don't know, like the first thing we will do will be to speak to our neuropediatrician and see how it can be linked. But I understand what you're saying. I'm just trying to understand how it works, and after we will see what is possible, of course. But so you don't remove all the drugs. You keep the drugs and you add.
Correct. It's an adjunctive therapy is what they say, but we're adding it on. So for the European community, I have been very open in sharing our data with European collaborators. In some countries, just our abstract and our poster was enough to convince the national health system to give it a try. So if we do see effect in some of the kids, we will share that quickly to make it more available.
Because our neuropediatrician is a referee in France for epilepsy, so so he should, I'm sure he's aware if we are speaking about that, because he's a national referee from France, so he should be aware of that. Okay, thank you.
Thank you so much, Christelle. That was such a great question, and thank you so much, Dr. Grinspan, for being on the call and sharing all of that information with us.
My pleasure.
Take care, and we'll talk to you soon. Bye.